Assalamu alaikum. My name is Aylin. I'm a second year medical student, and today I'm going to be talking about glycogen metabolism. Inshallah, it's going to be a very easy and short lecture, so let's just get into it. I'm going to be talking briefly about the terminology that you're going to encounter in the next few lectures, and in this lecture, which are included in gluconeogenesis, glycogenesis, and glycolysis. And then I'm going to be talking about glycogen synthesis and metabolism, and then how your body regulates the glycogen synthesis and metabolism. And then at the end, there's a few diseases that affect the glycogen storage in your body. So we're just going to be mentioning that. To begin with, these are the words that you're going to encounter, and they all look very, very similar. So I just tried to put it in a table that you guys can revise from, so you don't get confused. Basically, glucose, what do you guys know about glucose? It is the simple sugar that your body uses for energy. And then when your body is fulfilled with the energy that it has, it begins to store the glucose as glycogen, okay? And then when your body needs the glycogen, it's going to degrade it, which is glycogenolysis. And then when your body is storing it as glycogen, the process is glycogenesis. So I don't want to get into detail with this because this is not exactly my lecture, but you guys can just use this as revision. And also here, this was mentioned in your slides, and you guys can see that glucose itself can get turned into glycogen in the process of glycogenesis and then degraded again into glycogen, into glucose, but the process of glycogenolysis, and then you guys know glycolysis. Okay, so first of all, we have glucose. I said that it's the basic sugar that your body uses for energy. But where do we get this glucose from? First of all, the main glucose intake is from exogenous sources. Like for example, you woke up in the morning and you ate breakfast, okay? Your glucose is going to be very, very high. And then as the day goes by, it's going to decrease, okay? Just to say that you just ate breakfast and you didn't eat anything after. It's only one meal, okay? It decreases. And about 6 hours after you eat, the glucose that you got from your meal, it's going to just disappear. You're going to use it. You're going to turn it into energy, okay? And then you're hungry, but you're not going to eat. So what is your body going to do? It's going to use the glycogen stores. What is glycogen? I said that glycogen is the storage form of glucose. Like your body can save this for when it's fasting, when it's starving. Like it's just a mechanism that your body does for survival, okay? You're going to use the glycogen. And then glycogen, it has stores, obviously. It's not going to last you forever. And then around the end of the day and then towards the next day, if you still didn't eat, your body is going to make its own glucose, which is called gluconeogenesis. You're going to have a whole lecture on this, so I'm not going to go into detail about this. Okay. What is glycogen? As I said, it's a storage form of glucose, and it is mainly stored in the muscles and liver. The muscles and liver, they have one key difference that you guys have to know, is that is everything okay? Okay. So in the liver, you have the enzyme glucose 6-phosphatase, which breaks down glucose 6-phosphate into glucose, which leaves through the blood. But in your muscles, you don't have glucose 6-phosphatase, and therefore it does not leave to the blood. So when we're thinking about the glucose that's in your blood, that's coming from the glycogen, it's only coming from your liver, not from your muscles. Your muscles only use the glycogen for itself. Like for example, when you're exercising, when you're running, your muscles need energy, and they get this energy, if you haven't eaten, for example, from glycogen, okay? And it doesn't contribute to the blood glucose. I'm going to talk about this in detail, but this is just what was mentioned in your slides. Okay, so glycogen is just a bunch of glucose molecules stuck together, but they're not all just clumped up, okay? It's mainly a linear chain. As you can see, it's just like a straight chain. And then in some places, you have branching, okay? Branching. And what contributes to this branching? Basically, it's just how where the bond is on the molecule. Like for example, when you're saying alpha 1 to 4 bond, that means it's the carbon 1 stuck to the carbon 4 of the other molecule. So this is 1, this is 4, okay? And here this is 1, this is 4. So this is a linear molecule, linear shape, okay? And then here it's 1 to 6, which differs in where this is one glucose is going to stick here or here, you know, it just differs. It's just the name of the carbon, the number of the carbon. So this is 1, This is 6. And this is the branched chain. So the linear is 1 to 4. The branched is 1 to 6. Okay? Is everything clear? Okay. So I talked about glucose, glycogen, what glycogen is, why does our body store glycogen. But then sometimes when you, as I said, fasting or starving, your body wants to use the energy or the glucose that it stored. This process is called glycogenesis, okay? Oh, never mind. Glycogenesis is when you're making the glucose, because genesis means the formation of. So glycogenesis, you're forming glycogen, okay? My bad. So there's a bunch of steps to this, and I'm gonna go into detail in them. The main idea that you guys have to know is that it shifts to the formation of glycogen because your body is at a high energy, okay? So the glucose 6-phosphate levels in your body are high. So what is glucose 6-phosphate? Glucose 6-phosphate is basically, you guys studied glycolysis. So you have glucose, and then it goes inside the cell. Inside the cell, it gets phosphorylated. Because it gets phosphorylated, it can't escape. And the phosphorylated form of glucose is glucose 6-phosphate. So when I'm saying glucose 6-phosphate, I'm just basically saying glucose, okay? So when the glucose levels in your body are high, your body shifts to store the glucose in the form of glycogen because you can't afford to lose the energy. Halas, you already have so much energy, you just want to store it. You just want to keep it down, okay? However, a doctor mentioned this, that your glycogen stores are not endless. Like you can't keep storing glucose. And if you keep eating, it's not just going to get stored forever. So this is why some people gain weight, because the glycogen stores get filled up. So the glucose is going to get converted to body fat and it's going to accumulate in your body, okay? So glucose gets, okay, this is just the equation. I'm going to be talking about it in detail, about like how these specific metabolites form. Okay, so now we're talking about glycogenesis, which is the formation of glycogen synthesis. Okay, I said there's glucose here, and then it enters the cell, and then it gets phosphorylated to glucose 6-phosphate. Okay, glucose 6-phosphate gets converted to glucose 1-phosphate using the enzyme phosphoglucomutase. Okay, this is the enzyme, and it's reversible. Everything I'm saying is written here, but because I want to use the same exact picture Dr. Abdul used, so you guys don't get confused. Okay, this is the first enzyme that's in the glycogen synthesis, phosphoglucomutase. It converts glucose 6-phosphate to glucose 1-phosphate. And then the next step, which is probably one of the most important steps in this reaction, is the formation of UDP glucose. Why do we form UDP glucose? Because this is the active form of glucose. This is the glucose that can then form glycogen, okay? It has to have a UDP group. And where does it get it from? From UTP, okay? So UTP plus glucose 1-phosphate, which is from glucose 6-phosphate, gets converted to UDP glucose using the enzyme UDP glucose pyrophosphorylase, okay? UDP glucose pyrophosphorylase. It forms UDP glucose. We need UDP glucose because it's the active form, and this is what can help us form the glycogen, okay? Another important thing that I want to mention here is glycogenin. Glycogenin is the core protein. And if, for example, you have glucose inside the cell, they're not just going to accumulate and form and just, I don't know, aggregate with each other. There has to be like a core thing, like you're building something. There has to be something that you're building upon. So this is what glycogenin is. It's just the molecule or the protein that is responsible for giving the first glucose. So khalas, the glycogen gets synthesized and it gets the glucose just gets built upon it, So then we have UDP glucose, and we said this is the active form of glucose that's used in the formation of glycogen, okay? And then through the enzyme glycogen synthase, it forms the linear glycogen. What was the linear glycogen? It was the 1 to 4 bonds, okay? But what we said that glycogen is not only a linear molecule, it's also a branched molecule, okay? So we have an enzyme called branching enzyme, okay? Branching enzyme is the enzyme that forms the alpha 1 to 6 bonds, which is what? The branched glycogen, okay? I'm just gonna go over these again because I wanna draw something for you guys. So we said the first reaction is glucose 6-phosphate to glucose 1-phosphate, and it's through the enzyme phosphoglucomutase. Okay, glucose 6-phosphate to glucose 1-phosphate. Okay, glucose 1-phosphate with the addition of UTP gets converted to UDP glucose. Okay, UDP glucose. And what is UDP glucose? It's the active form of glucose. Okay, and then when we have the active form of glucose, خلاص, we can synthesize glycogen, and we can do the linear chain, we can do the branch chain, we can do whatever we want if we have the enzymes that we need. And what are the enzymes that we need? Glycogen synthase, which is the, what did you guys remember from the glycogen synthase? It's the alpha 1 to 4 or 1 to 6 bonds. It's the 1 to 4, the linear, okay? And then we said that glycogen is not only linear, it is branched. So we have the enzyme, the branching enzyme. But also in the slides, there is the amylo alpha 1 to 4, 1 to 6 trans glucosidase enzyme. I don't know, Dr. Amjbar mentioned it. So I think it is important to know the amylo alpha 1 to 4, 1 to 6 trans glucosidase. This is basically the branching enzyme. It's just a fancy name for the branching enzyme. And it's the one that forms the branch structure of glycogen, okay? And then there was another enzyme that was mentioned in the slides, which is not related to this picture here because it's just like a concept. It's a nucleoside diphosphate kinase. So we said that UTP plus glucose 1-phosphate turns into UDP glucose, okay? So when the glucose gets used, the UDP, what is it going to do? It has no function in the cell, okay? So because we don't want to waste it, it gets converted back to UTP so it can get recycled. So this is the enzyme nucleoside diphosphate kinase. It's the one that recycles UDP from UDP glucose, and then it can get used again, all over again, okay? Is everything clear so far? I'm just going to go over it really, really fast. So basically you have glucose 6-phosphate gets converted to glucose 1-phosphate using which enzyme? Okay, phosphoglucomutase. It converts glucose 6-phosphate to glucose 1-phosphate. And what do we mean by 6-phosphate, 1-phosphate? It's just the place where the phosphate is added. Like, is it at the 1 carbon, 6 carbon, it's just a name. And then the important step here is the converting glucose 1-phosphate to UDP glucose, because it's the active form of glucose that can be used to form glycogen, okay? We use the enzyme UDP glucose pyrophosphorylase, okay? And then when we have UDP glucose, خلاص, we're done. We can easily form the glycogen, but we have the linear chain and then the branch chain. So we have two enzymes. We have the glycogen synthase and the branching enzyme, okay? This is it for glycogen synthesis. Do you guys have any questions? Because I'm going to move on to glycogen breakdown. If you guys are typing something in the chat, let me know because I can't see from here, okay? Okay, I'm going to take this as a yes and move on to glycogenolysis, which is the breakdown of glycogen. Now we're saying that you are stranded in the desert, you just had breakfast, you can't eat anything anymore, but your body needs to utilize energy. Your body needs the glucose. So you break down the glycogen stores, and we said that we store the glycogen in the liver and the muscles. So it makes sense to say that the breakdown of glycogen happens in the liver and the muscles, okay? So we have these 3 enzymes. I'm not going to say them now because I'm just going to go over and talk about them. It's just for your information, like just for the slide. Okay, we have glycogen, which is where? In the muscle and the liver, okay? This glycogen gets converted to glucose 1-phosphate. It's basically, it's not the reverse of storing glycogen because there's different enzymes. That's a very important concept to keep in mind. Like, you're not just reversing the steps because it's a different set of enzymes, okay? It's a whole different process. So glycogen has linear chain, branch chain. Linear 1 to 4, branched 1 to 6. So it makes sense to say that because we needed 2 enzymes to form glycogen, we need 2 enzymes to break down glycogen. These two enzymes are phosphorylase and debranching enzyme. And phosphorylase is for the 1 to 4 like links, the linear chain, and debranching enzyme is for what? As the name indicates, for the branches, 1 to 6, okay? When we break down the glycogen and the linear molecules and the branch molecules, we get glucose 1-phosphate. And then glucose 1-phosphate through the enzyme phosphoglucomutase gets converted to glucose 6-phosphate. This was, I don't know if you remember this, but this was the reversible reaction that was in the beginning here. This was there's 2 arrows, that means it's reversible. So this is the only reversible reaction. You're converting glucose 1-phosphate to glucose 6-phosphate by phosphoglucomutase, and in the synthesis of glycogen, you're converting glucose 6-phosphate to glucose 1-phosphate by phosphoglucomutase 2. Okay. I added some text because this is what I want to mention, but because I didn't want to overwhelm the slide. So here I said that the phosphorylase is 1 to 4 cleavage, which is the linear chain, and it doesn't require ATP, but it requires a phosphate group. Okay, and glucose 1-phosphate gets trapped inside the cell. What did we talked about this? When you phosphorylate glucose, it gets trapped inside the cell. So basically, when you're donating a phosphate group to when you're breaking it down, you're trapping it inside the cell. So glucose 1-phosphate is inside the cell, and it was mentioned in the slides that it requires vitamin B6. Okay, for the debranching enzyme, we said that it was from the 1 to 6 like chain, which is the branches, and then there's this really, really important concept that I want you guys to focus on. Basically, this is glycogen. Okay, let's just say this is the branch. Okay, this is the linear, this is the branch. Okay, so we said that the phosphorylase cuts the linear chain. Okay, it just cuts here, 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 and then it reaches the branches, and it just doesn't know what to do. It just pauses. Okay, it doesn't have the ability to cut past it. So this is where the debranching enzyme comes into place. Basically, what it does is that it takes all of these and it puts them here. So it adds them here so the phosphorylase can complete its process. But as you guys saw, there's one here that it didn't take. There's one glucose that it was still hanging. So after it takes it, it's like this. It's a very, very long chain. And then there's one extra glycogen here. And this is the glycogen, the glucose. It's all glycogen, but I'm talking about the specific molecule, which is the glucose. This one gets left behind, okay? But then, as I said, the phosphorylase is not going to be able to continue. So there's this part of the debranching enzyme that's called glycosidase, which takes this one and releases it as free glucose, okay? So we're converting glycogen to glucose 1-phosphate. So all of this is going to turn into glucose 1-phosphate, okay? Which later on undergoes glycolysis or the steps that it requires it to actually form glucose. But then you have this one alone piece of glucose which is attached to the glycogen, which I'm just gonna call it glucose because it's alone and it's just gonna get sent off to the bloodstream as the only glucose molecule. Okay? Did you guys get this point? It's really important. That we have the phosphorylase cuts everything in the linear chain, and then when it reaches a branch, it just pauses, and then the debranching enzyme comes and takes, for example, let's say we have 5 that are branched off. It takes 4, okay? It takes 4 and it puts them down, up. It puts them where the phosphorylase is going to complete its process. But then there's one left. What is this one? What is it for? This one left is the one that's going to be cleaved by the glycosidase, and it's going to be sent to the bloodstream as free glucose. So when we're breaking down glycogen, we get a lot of glucose 1-phosphate and 1 free glucose from each branch we're cutting. Okay, does that make sense? Is everything clear?
Yes.
Okay, I'm gonna move on. So when you get glucose 6-phosphate, which is after converting glucose 1-phosphate to glucose 6-phosphate, it's gonna undergo glycolysis. You guys know the steps of glycolysis. Glucose 6-phosphate, it's trapped inside the cell, and then later on, at the end of the glycolysis, you're gonna form pyruvate, and then you're gonna get energy, ATP, whatever. Okay, so this is one fate of the glucose 6-phosphate of the breakdown of glycogen undergoing glycolysis. Or the glucose 6-phosphate can be transported into the liver endoplasmic reticulum. Okay. What is the enzyme that takes this glucose 6-phosphate to the liver and endoplasmic reticulum? It's a glucose 6-phosphate translocase. From the name, it's like translocating the glucose 6-phosphate. It's taking it to the endoplasmic reticulum. Like this process could be taking place in the liver, but we want the specific, the body decides the specific glucose 6-phosphate. I don't want it to undergo glycolysis. I want it to immediately get turned into glucose. Okay, so it takes it to the endoplasmic reticulum because it has an enzyme that's called glucose 6-phosphatase. This is the enzyme we talked about in the beginning of the lecture. This is the enzyme that's not present in the muscles. Okay, this is the enzyme that's only present in the liver. It's present in the liver. What does it do? I said that it converts glucose 6-phosphate to free glucose. So the liver contributes the free glucose that's in your bloodstream. So phosphatase, as the name implies, it's removing a phosphate group from glucose 6-phosphate. So when you're removing a phosphate group, khalas, you're just left with glucose. And glucose can be easily transported outside the cells through what transporters? What are the glucose transporters called? You guys know? Okay, they're called the blood transporters. These transporters, if the glucose is phosphorylated, it just doesn't let it out. But because now we're removing the phosphate, the phosphatase, the glucose is going to be free. It's just going to leave the cell, okay? And then it's going to go into the bloodstream and it's going to accumulate in your blood and it's going to account for the glucose that's in your blood. So as I mentioned, the muscle doesn't have glucose 6-phosphatase. So the muscle is selfish. It just wants to use the glucose for its own good. You know, you're exercising, you're running, you're going through like a fight or flight response. The glucose just wants to use it on its own, okay? But it doesn't break it, like it doesn't break the phosphate down, so it just can't leave. It just stays inside the muscles. So it doesn't have glucose 6-phosphatase, okay? Now let's go to question. Does anyone want to read? Okay, I'm gonna read. The immediate degradation of glycogen under normal conditions gives rise to which of the following? A, more glucose than glucose 1-phosphate. B, more glucose 1-phosphate than glucose. C, equal amounts of glucose and glucose 1-phosphate. D, neither glucose nor glucose 1-phosphate. And E, only glucose 1-phosphate. I'm gonna give you some time to think about it. And if you guys wrote something in the chat, let me know because I can't say it to me. Do you guys have anything to say? Okay, so the question is basically asking when you're degrading glycogen, what do you get? Like when you're breaking down glycogen. Does anyone say anything in the chat? Okay, I'm gonna move on. The answer is B. More glucose 1-phosphate than normal glucose. Because remember the enzymes, the one free glucose that gets transported, it's only one, and the rest are all glucose 1-phosphate. So the question here is just basically asking you that what is the product of glycogenolysis, which is glucose 1-phosphate and 1 glucose per thing that you break down. Okay, do you guys have any other questions? Because I'm gonna move on to regulations. Okay, I'm gonna take that as a no. Okay, so I was saying you make glycogen, you degrade glycogen, you turn it into glucose, your body stores glycogen, then you need the glycogen. This is all happening like beyond your control. And obviously there has to be some sort of regulation that's going on in your body that's telling your body, oh, I need more energy. oh, I have enough energy. خلاص, I'm gonna stay in my. For example, if you just ate, your body خلاص doesn't need more energy. It's fulfilled. Or when you're starving, for example. So there are three mechanisms in which your body can regulate these processes, and basically it regulates the enzymes, the main enzymes. Okay. So first of all, we have hormones. We have glucagon, insulin, and epinephrine. These are the three most important hormones I'm going to talk about. Then we have allosteric regulation, which is through calmodulin, calcium, ATP, and AMP. Calmodulin and calcium are both when your muscle is like, when your muscle wants to contract, for example. And ATP and AMP are like, ATP is like high energy, AMP low energy. So this is what it means by allosteric. And for example, ATP gets converted, like it's formed when you're going through, for example, like the Krebs cycle. So when you have a lot of ATP is gonna go back to the enzymes and tell it, خلاص, stop, I don't want more. But it's like allosterically regulating the enzymes. It's like the end product of a reaction going back to one of the enzymes and telling it to either do the job faster or slow down. Okay. And then we have covalent regulation, which is when you phosphorylate a molecule, you add a kinase to it. The kinase is going to add a phosphate group. Okay. So the most important concept that I think if you really understand this, the lecture is going to be really easy to you, is the fact that certain enzymes work or don't work based on if they're phosphorylated or dephosphorylated. Dephosphorylated means they're not phosphorylated. So some enzymes, they are active when they're phosphorylated. Some enzymes are active when they're dephosphorylated. For knowing this, you can like understand how the body regulates the most important enzymes in making glycogen and degrading glycogen. Okay, so first enzyme I'm going to talk about is glycogen synthase. What was this enzyme for the formation of glycogen or the breakdown of glycogen? Okay, glycogen synthase was for the formation of glycogen, okay? And when the enzyme is phosphorylated, it's inactive. Like for example, if the kinase goes inside and adds a phosphate group on this enzyme, it's just gonna stop working, okay? On the other hand, when the phosphate group gets dephosphorylated, it's getting away from the enzyme, it's active, it's working, okay? On the other hand, we have glycogen phosphorylase. This enzyme is responsible for degrading the glycogen, okay? And this enzyme loves to be phosphorylated. I don't know, just remember it, it's phosphorylase, it loves to be phosphorylated. That means it's only going to work when you add a phosphate group to it. But then when you remove the phosphate group, it's gonna stop working, it's inactive, okay? So this is the key idea that's going to be built upon when I'm talking about the hormones, for example. So do you guys understand this? Please say yes. Okay, I'm taking this as a yes, and I'm gonna move on. Okay. I said that the enzymes can be regulated through hormones, glucagon, epinephrine, and insulin. Okay. Okay, glucagon. Yeah, glucagon, epinephrine, and insulin. The first two I'm going to talk about are glucagon and epinephrine because they somehow have the same like mechanism of action. Okay, they both have the same effect. So here we have glycogen to glucose. The arrow is pointing down. So we're making glucose. We're degrading glycogen. What is the enzyme? Glycogen phosphorylase. Okay, and we said that phosphorylase loves to be phosphorylated, and it's gonna be active when you add a phosphate group to it. So you can see here the P, the phosphate group, it's on the phosphorylase. That means it's working. You're converting glycogen to glucose. On the other hand, when you look here, glycogen synthase, it is phosphorylated. But what did we say? When glycogen synthase is phosphorylated, it doesn't work. So you're not gonna convert glucose to glycogen. You're not gonna make glycogen, okay? So the only process taking place is glycogen phosphorylase, okay? And this is through the effects of glucagon and epinephrine. So let's see how they do it. So first of all, what is glucagon? What is epinephrine? So glucagon is a hormone that's released by your pancreas when you're fasting, for example. It signals low glucose in your blood. So what do you want to do? You want to break down the glycogen. You want to increase your blood glucose. So it acts on the enzymes. Epinephrine on the other hand, I think you guys know epinephrine. It's when, for example, you're running away from like a bear, or like you're stressed, for example, your body wants energy, needs energy. Okay, it's like I told you, it's like something that was like evolutionary. Like, for example, people when they long time ago, when they were running away from bears, they needed the energy to run. Okay, so epinephrine gets produced, and it utilizes every ounce of energy that it can find in your body. So how does it do that? Here in the picture, you see you have epinephrine and glucagon. They both act on adenine cyclase. You guys don't really have to know this. The thing you have to know is that it increases cyclic AMP. Cyclic AMP is just a messenger, it's just a signaling pathway. It's a messenger that increases pKa. What is pKa? Protein kinase A. From the name, kinase, it adds a phosphate group. So it goes to both the enzymes, phosphorylase and the phosphorylase. In the picture, it should be synthase, okay? The doctor explained, it goes to the phosphorylase and glycogen synthase, and it phosphorylates both of them. When you do that, which one's the one active? If you're phosphorylating glycogen phosphorylase and glycogen synthase, which one's the one that's going to work? Because you have them both, you always have them both, but it's just a mechanism of which one works, which one doesn't work. So when you're phosphorylating them, when you're increasing pKa, protein kinase A, which adds a phosphate group on the enzymes, the phosphorylase is going to work, which means you're going to break down the glycogen. You're going to increase the glucose levels in your body, and that's what you want. This is what these hormones are looking for in your body. They want to increase the glucose levels in your body. Okay, so they phosphorylate both the enzymes, because one works when it's phosphorylated, one doesn't work. Okay, is this clear? I wrote for you guys here if you want to read it. It's exactly what I said, and it's just that you have to know that through the signaling pathway of cyclic AMP, pKa increases where it gets formed, and then it phosphorylates glycogen synthase and glycogen phosphorylase, which leads to the breakdown of glycogen. Okay, moving on. This is the same exact concept, but there's an extra thing that the doctor mentioned here, so I'm going to talk about it. When your muscles are contracting here, they release calcium or calmodulin. Okay, calcium doesn't work through the cyclic AMP mechanism, but itself calcium goes and activates phosphorylase kinase, which is equivalent to pKa, protein kinase A, and whenever you see kinase, just think of phosphate, think of energy. Okay, it activates glycogen phosphorylase, which is the one that converts glycogen to glucose. Okay, so when your muscles are contracting, they want energy. Okay, so yeah, through calcium and calmodulin, you activate phosphorylase kinase, which is, like I told you, equivalent to pKa, and it activates glycogen phosphorylase. Okay, that was the hormonal. Okay, a small little part I'm going to talk about is the allosteric. I told you, like the end product of I don't know, like for example, you form ATP. ATP increases. ATP itself is going to go back to one of the enzymes and tells it, okay, stop. We have enough ATP. This is the allosteric regulation that I'm talking about. So here we're saying glycogen, glucose, phosphorylase degrades glycogen, synthase makes or builds glycogen. So here we have glucose 6-phosphate. What was glucose 6-phosphate? It was the glucose that's in your body. So when you eat, your glucose 6-phosphate is going to increase. So when it increases, your body deviates and stores it. Okay, so the green indicates it's a positive feedback. It activates glycogen synthase because you want to store the glucose. You don't want, your body has enough glucose, it wants to store it. Okay, it doesn't want to use the energy anymore right now, it wants to use it later. okay? So glucose 6-phosphate, which is basically just glucose. Think about it. And when you have خلاص, enough glucose, you can't have more. Your body can't tolerate more. So it positively increases activates glycogen synthase. On the other hand, when you have ATP, this is supposed to be something else, but because it got cut, ignore this. I'm talking about ATP and AMP. ATP is signaling high energy in your body. AMP is signaling low energy in your body. So when you have a lot of ATP, it's going to negatively activate phosphorylase. يعني it's not gonna activate خلاص, it's gonna make it stop working because you already have a lot of energy. Why would you want to convert glycogen to glucose? Glucose is energy basically. So when you have a lot of energy, it's enough. Your body it doesn't want more. So it stays as glycogen. But it doesn't activate phosphorylase. And what was phosphorylase? The enzyme responsible for glycogen metabolism, okay? And the degradation of glycogen. On the other hand, when you're at a low energy, AMP is signaling low energy, your body wants glucose. It wants to degrade the glycogen. You want energy. As you see here, green, it activates glycogen phosphorylase, okay? That is all what you have to know for the allosteric regulation. Yeah, just think about it logically. Like you have energy, your body doesn't want more energy. خلاص, it's gonna activate the enzymes responsible for storing glucose, not degrading glucose, because you don't want glucose anymore, you don't want more energy. And on the other hand, it's like if you're at a low energy, your body wants glucose, it wants energy, it degrades glycogen. Is that clear? Okay. Moving on, we talked about glucagon and epinephrine. These hormones increase your blood glucose levels, okay? They increase it. Why? How do they increase it? They activate glycogen phosphorylase. They add a phosphate group to it, and that makes it active. They also add a phosphate group to glycogen synthase, which inactivates it, okay? On the other hand, we have insulin. I think you guys took insulin. It's the hormone released also by your pancreas when you just ate. It's signaling, خلاص, I am fed, I don't want more energy, I am fulfilled, okay? So it's going to activate glycogen synthase because you want to store the glycogen. You don't want more energy. You already just ate, your body doesn't want more energy. So what did we say? That glycogen synthase is active when it's phosphorylated or dephosphorylated? Glycogen synthase is active when it's dephosphorylated, okay? So what does insulin do? Let's go to the next slide. Insulin binds to its receptor. This is the receptor for insulin. And then it activates tyrosine kinase, okay? Tyrosine kinase, in return, activates protein phosphatase. What did we say about phosphatase? Anything phosphatase. The ASE at the end. That means it's breaking down, cutting something. It's removing the phosphatase. So it goes to glycogen synthase and it removes the phosphate. So we end up with glycogen synthase that's dephosphorylated. It doesn't have a phosphate. So what does that mean? It's active, it's working. And when glycogen synthase is working, you're converting glucose to glycogen because your body is fulfilled. It has enough energy. It's good. Okay? Is that clear? I just put this for you guys as like a test for I want you to tell me which one's going to be activated. In here we said tyrosine kinase, it's for insulin. And what does insulin do? Insulin signals that I'm not hungry anymore, I'm done, I'm fed. So I want to store the glucose I just got in. I don't want to use it anymore. So it activates phosphatase, because what does phosphatase do? Remove the phosphate group off of glycogen synthase. So what do you end up with? Yes, glycogen synthesis. Yes, synthesis. Glycogen synthesis, because you don't want more glucose. You want to synthesize glycogen. Okay. On the other hand, here, epinephrine and glucagon. They activate cyclic AMP, which activates pKa. Okay, pKa is going to add a phosphate group to glycogen phosphorylase and glycogen synthase. But we said that glycogen synthase is inactivated when it's phosphorylated, so it's not going to work. What's going to work is glycogen phosphorylase, which is going to break down glycogen into glucose. Okay, this is it here. Is this concept clear for everyone? Okay. I'm gonna move on. Can someone read this question or do I read it? Okay, regulation of glycogen metabolism is tightly controlled at the level of the activity of glycogen phosphorylase. Which of the following is known to act as a negative effector of the phosphorylated, which is the most active form of glycogen phosphorylase? What do you guys think? Okay, what they're saying here is that glycogen phosphorylase is active when it's phosphorylated, when you add a phosphate group to it, okay? So they're telling you which one is the negative effector, which one inhibits this enzyme. We don't want this enzyme to work, okay? So let's go through the options one by one. So phosphorylase is basically when you're breaking down glycogen phosphorylase. Where? I want you guys to answer. Okay, so the answer is glucose 6-phosphate. Glucose 6-phosphate is what? It's glucose, okay? So when you already have glucose, it's gonna go and negatively inhibit this enzyme. Why? Because this enzyme is responsible for the breakdown of glycogen to glucose, okay? And when you already have glucose, you don't want more. So it's gonna negatively or like make the enzyme not work. And AMP is signaling low energy, okay? So it doesn't negatively inhibit this enzyme. بالعكس, it wants it to work. It wants it to increase glucose. So this is wrong. Cyclic AMP is just a mechanism of the signaling pathway, so it doesn't have to do with it's not a covalent or a hormonal, or it doesn't regulate the enzyme. Calcium is also wrong because phosphorylase, it's also, it doesn't act as a negative effector, okay? It's not, I don't know, it doesn't act as a negative effector. It doesn't want this enzyme to stop working. It wants it to keep on working because you want glucose in your muscles, okay? And also protein kinase A is the same reasoning as cyclic AMP, because it's just a mechanism of how what's it called? The signaling goes, okay? Okay, now I'm gonna move on to the last topic of this lecture, which are the diseases. But do you guys have any questions first? Okay, so there's only 2 diseases or disorders that I'm going to talk about, and they're very, very simple, okay? And they always come in MCQs, even SQs. So please make sure you know them, okay? The first one is McArdle disease or glycogen storage disease type 5. So it's classified as myophosphorylase deficiency. And what did we say? What is phosphorylase? It converts glycogen to glucose. And myo just means it's in the muscles, okay? So this is the this is the enzyme that converts glycogen to glucose in the muscles, okay? So when you have a deficiency in this enzyme, your muscles are not going to be able to utilize the glycogen that you have. It's not going to be able to degrade the glycogen, okay? So deficiency in this enzyme leads to, as I said, decreased breakdown of glycogen during exercise, which causes poor exercise tolerance, and you don't have energy, you can't work, you can't walk, you can't even work out. People with this disease have trouble like even walking, okay? Muscle pain, cramps, and rhabdomyolysis, which is basically just your muscles getting degraded. and your muscles are getting so weak, they're getting degraded. And I put this urine sample because it causes kidney problems, because the muscles are getting degraded and the components of the muscles are going and getting filtered in your kidneys, and your kidneys just can't take it anymore, okay? And what do you do with patients with this disease? You give them glucose, because they cannot degrade their own glucose, so you have to give them exogenous glucose the entire time, okay? Next disease is Pompe's disease, and as you can see here, there's a baby, because the effects come on very, very early on in life, okay? So what exactly is this disease? So there's this really important concept that glycogen can get broken down in the lysosome by the enzyme alpha-1 to 4 glucosidase, which is also called acid maltase. So glycogen, it gets broken down in the lysosome by this enzyme. So thinking about it, if you have a deficiency in this enzyme, the glycogen is gonna accumulate. It can't get degraded in the lysosome. But it's gonna accumulate, and it's gonna form like in the vacuole, in the cytosol of the cell, the cytosol, which is like the free fluid, it's gonna form vacuoles. Vacuoles of what? Vacuoles of glycogen, because you cannot break it down. So the manifestations are like very random. They're just like heart, respiratory, feeding problems, but just know that it affects infants, okay? And it's glucosidase enzyme. That is the most important part of this disease, okay? That is it for this lecture. Do you guys have any questions? If you guys have any questions, please don't hesitate to let me know and text me. I'm always willing to help you out. But don't forget to scan this code. So, thank you so much.